Our next speaker is Elizaveta Taranenko, and I hope I didn't trash your name too bad, Lisa. She earned a master's in Genomics and Bioinformatics at Siberian Federal University. So she is currently a PhD student in plant physiology. Her PhD thesis is about plant genomics, epigenetics, and genome annotation of oil palm. Wow. So she's interested in all kinds of areas, loves working on mammals, studying genomics and transcriptomics. Another fascinating type of omics data is metagenomics. And we have been hearing about all the microbiome a lot, its role in health, disease, therapy, and even prevention. And now let's invite Lisa from the Tauber Bioinformatics Research Center to speak about metagenomics, data analysis, and how it can be performed in the T-BioInfo platform. So welcome, Lisa. We really are looking forward to hearing about your area of interest. The floor is yours.
Thank you for having me. Can you see my screen well? Yep. Okay. Yep. My name is Lisa. Very happy to be here, and I'll be speaking about QIIME. It is a tool that we use to analyze metagenomics data. And metagenomics is really about microbial communities. We want to learn the composition of them and compare them between each other. Metagenomics is used in a variety of areas in agriculture, healthcare, biotechnology, everywhere. But this topic will be mostly about technical side of analysis on the platform, not the biological one. So on our platform, there is a possibility to use two types of data. Upload data. There is a manual upload when you have all the files downloaded on your laptop or computer, and NCBI upload. So the set of files needed for each is a bit different. In each case, you need metadata where you have the groupings of your interest. But for manual upload, you also have to have FASTQ files with your reads. And in case it is a multiplex data, you also need barcodes. So maybe you're already familiar with the concept of multiplex data. It's when in one run we have several samples and they're differentiated by the barcodes. So when this file is in QIIME, it needs these barcodes to separate the samples again. And in NCBI, we only need metadata, and this metadata is used to download all the FASTQ files that we need. So on the upload panel, you have to pick your type of upload, manual or NCBI, and you also have to pick if you have single or paired end reads, and select the file type if it's manual upload. For NCBI upload, this is a typical metadata file, and the critical column here is the first one, which has the SRA IDs, and they will be used to download all the data you need. Where to find this file? So you have to search the SRA archive and look for the study that interests you, and there will be this metadata button that will start the download of the metadata file. Just be careful because sometimes it downloads the file with commas as separators, but we can only accept tab separated files. So sometimes this file needs a bit of correcting before running the pipeline. So when the upload is done either manually or with the help of NCBI, we can start building our graph. And QIIME is separated into 5 subsections, and we start with pre-processing. So first we import data to QIIME, then we filter samples. We can filter out samples that are too few in the number of reads. And then we want to see the quality control, the quality of our reads. Then moving on to processing OTUs, that's operational taxonomic units. And here we have two algorithms, denoising algorithm, data2 and deblur. And what they do, they create the, they identify the operational taxonomic units and they calculate the abundance in each sample. And with this table, we can proceed to taxonomy, where we classify our OTUs with existing database. In this case, it's Silva database for 16S RNA, where you have to pick your region of the region that was used for sequencing. In our case, it was variable region 4. And you can alter your classification, resulting data if you want. And then we can move on to metrics, where core metrics calculates all the alpha and beta diversity indexes. And then we can visualize alpha diversity with box plots. And for beta diversity, we will need to pick a column for testing beta diversity. And here I want to remind what beta diversity is about, because for alpha diversity, this is different measures that study the community within itself. But beta diversity compares the communities between each other. So here we have to pick some column that has a grouping of samples that we're interested in. In my case, it was the body site that the sample was taken from. And after beta diversity, we also can use interactive visualization. And there we also have to pick two columns to visualize. In my case, I picked body site and antibiotic usage. So when we are ready with our table and graph, and we ran this pipeline and it's finished successfully, you will see this external dashboard here. But actually, let me try and go to the browser. Okay, it's here. So this is the pipeline that successfully finished, and I want to check my results with the dashboard. So let's go here. You can see that in the dashboard, there are 3 areas of data. There's big bar plots, some NMDS, and alpha diversity box plots. And apart from this dashboard tab, we also have a table tab. This is the table that all our plots are based on. And you can have your OTU IDs and all the counts here for each sample. This is rarefied table, meaning that if you sum up all the abundances for each sample, they will be the same. You can download this data and it will open you the raw look of this file. You can just copy it and paste it into text document. You can also download metadata or taxonomy, where for taxonomy it's the same logic. You have your OTU ID and all the taxonomic units. So, and also you can look what this OTU is associated with, what taxonomic, what taxa it is associated with. Here we have family, but again, you can look at different levels. So back to dashboard. This rarefied table is used to plot these bar plots. Let me make it a bit bigger. These bar plots that have samples and relative abundance, and you can play with this graph the way you want. You can, again, pick the different taxonomic levels. You can pick different coloring to your liking. Oh, this is not very good. Okay, let's stick to this. You can download a CSV of this table or anything. And if you're interested, for example, this sample what taxa it is and what exactly the abundance is for this sample, for this taxa. Next is an MDS. This is something like a principal component analysis or principal coordinate analysis, also a dimensional reduction technique that tries to show the pairwise dissimilarities between the samples. Here it's also colors are controlled by these columns in your metadata. And if I want not two, but only one, I can pick the same color here and it will be like one group. Also, if we point at it, it will show what is the symbol and the color and coordinates in the three-dimensional space and the sample name. And also the alpha diversity box plot. Here it only shows observed OTUs, and you can also group it by any column from your metadata. And for example, in body side, we have 71 observed OTUs in gut and 165 observed OTUs in palms, a lot more than in the gut. But this is the dashboard. But actually, QIIME creates a bit more information than that. And if you want to look into beta diversity or more indexes from alpha diversity, you can go to download pipeline output files. And here you will see a zip folder for each job that finished successfully. And I'm interested in alpha diversity and in beta diversity, for example. So let me go back to my presentation. Sorry about it. Okay, we already finished this. Yeah, so here you have to get familiar with how actually QIIME operates. It has two types of data. It has QZA files and QZV files, and both are normally unreadable to us with our regular tools. QZA has all the tables in it, and so we modify it into TXT tables for you to look at. But QZV files are visualization files, and they're specifically designed to be viewed on the online browser of QIIME. And it looks like this, and you just take the file with QZV at the end and drag it here, drop it. And for example, let's look at beta diversity. Here, Bray-Curtis distance is used to calculate the distance metrics for the samples, and we colored them the same way with body side. We can see nice differentiation between the body sides, but both palms are together, which is also makes sense. Another distance metric, Jacquard, is used here, and they both are visualized in the Emperor plot, which is principal coordinate analysis algorithm. And we have two more distance metrics that are used by QIIME. It's weighted unifractional and unweighted. Then if we move on to alpha diversity visualization, we can check out a bit more of the indices that there are. For example, faith index that shows us the evenness and richness of the community, but it takes into accordance the phylogenetic relationship between our OTUs. And here we again see that left palm and right palm are really rich compared to gut, but we actually can kind of see it in our bar plot. We can see that our gut samples, which are these, they are mostly comprised of three taxa. And for example, these are palms. We can see that there are a lot more different bacteria are there. So apart from box plots, QIIME also calculates statistics, all groups, and pairwise statistics. And for example, here's gut versus our 3 other tissues. And we can see that there is significant difference with palms, but it's not significantly different from tongue, which also makes sense. And other metrics are the evenness, which shows how even each OTU is present in the sample. And just remembering the box plot I showed you, the palms are a lot more evenly present than the gut, where we have only 3 bacteria there. And Shannon index, which also shows evenness and richness. So thank you very much for your time and for having me here. I'm ready for questions.
All right, I am here. I just have to switch toys to help you out here. We do have a couple questions, and thank you for all that, Lisa. That was fascinating. So here's the first question. How has metagenomic data analysis been used in cancer research?
I'm not sure about cancer research since I'm really never did it in cancer research. But I believe that just metagenomics in general for human is used excessively to understand some physiological problems that cannot be explained by other techniques. For example, there are some diseases that genetically we kind of understand, but still the behavior of our GI tract cannot be only explained by the genetics. That's when a metagenomics step in and try to analyze the microbiome in our gut and explain this behavior. But in cancer, I don't know how it's used there.
Okay. Sorry about that. It wasn't an attempt to throw you off. Hoping you had an answer there, but thank you. Yeah. What are major challenges faced during analyzing metagenomic data?
Well, we rely heavily on the database to classify our OTUs. So I think this is one of the hardest parts, because most of the databases, they don't have ideal taxonomic information. Sometimes we end up with order of the bacteria, and that's the best that we can get. This is a bit troublesome. And also there is a difficulty with understanding when there is a contamination, because contamination mostly is bacterial contamination, and here we already are working with bacteria. So sometimes it gets tricky, but I think that classification is kind of the hardest part in metagenomics.
Okay, thank you. How about this one? How can one transition from statistical analysis to interpretation of metagenomic data analysis?
From statistical analysis?
To interpretation of the metagenomic data.
You mean statistical analysis, not a major in statistical analysis, just how to go from this Kruskal-Wallis test to understand how it's biological. Okay.
We'll go with that.
Well, you have to work with biologists sometimes because, well, if you really know the subject well, I guess you can interpret it yourself. But most of the time, it really needs understanding of the processes and what we can expect, because a lot of things can go in a way that we do not expect, and that is wrong, because sometimes experiments go wrong, and we have to understand when we are wrong, where we're looking at something that doesn't make any sense. So I would pick a biologist in my team if I had a chance. And
phone a friend, right?
Sorry?
Phone a friend, right?
Yeah, something like that. Exactly.
Can microbial communities that you study be modulated with bacterial phages in a targeted way?
Sorry?
Can microbial communities that you study be modulated with bacteriophages in a targeted way?
We target them?
Yeah, bacteriophages. So can you target specific taxa of microbes with phages?
I'm not very knowledgeable in this area, but I think, yes, we can. But
and how would you identify such phages? Do you know?
Well, how we differentiate taxa by different differences in the genome in these variable parts. If we, I think that we can target our phages to these sequences, but I'm really not familiar with the process, how to do that.
Okay. How about this one? What are your thoughts on using the microbiome as a fertilizer in agriculture?
Well, I think I'm not sure, because there are different bacteria which work in the soil and which work in the gut. I'm not sure if the same bacteria will do the same job well. But we definitely use feces from other animals for soil. So maybe there is some idea in that.
Okay. Well, our apologies for putting you on the spot. We never know when the questions are going to come in and if they're all perfect for you. So thank you so much. And we will let you go for your afternoon. Thank you so much, Lisa. We really appreciate that. Yes,
Thank you. Bye.